Hello, and thanks for staying on. In the session yesterday that we had about clinical trials in the UK, we specifically skipped gene therapy and felt that it would be worth to dedicate specific sessions on gene therapy because it's one of the most talked topics in clinical research in Duchenne muscular dystrophy. And yesterday I thought it would be better to have a double act and wanted to do this today again with my colleague Francesco Montoni. Unfortunately, Francesco, who was planning to come, just had a health emergency this morning. Everything is okay, but he unfortunately can't join us. So I'll try to cover what the two of us were planning to present together alone. Now, the good news is that we will do some panel skipping this afternoon. So the session on gene therapy panel questions and answers at 2:30 is something we can probably skip because there will be plenty of time to ask questions in this session, in the following session, and Annemieke has said that she will be around as well if there are additional questions. I'm not entirely sure about Linda, but she may also be around. So if there are also questions to the basic researchers and about antisense, there will be opportunities to ask these questions. Now, just briefly following on from this slide that I had already introduced yesterday about the different phases of clinical trials. And there was a question then also, can't we open more sites, and can we avoid, or how can we avoid that patients and families need to travel? And I just wanted to show another graph. I often use this, even though it's more for common diseases, not for rare diseases. But I think it's a nice illustration about the complexity of drug development because it adds the boxes with the blue stripes. And what it shows is the more specific you come to drug testing, and once you go into the clinical phases, phase 2 and phase 3, it's actually the amount of costs, and that's what the costs reflect in the blue stripe boxes, increases as well. So if you test 10,000 compounds, it's very cheap. If you test one very robustly, it becomes extremely expensive, which to the parents that were in the room yesterday is one of the reasons why actually to increase this box is a challenge, even for big companies. Now, have all of you heard about viral vectors? Yes. So we had a brilliant session this morning from Annemieke and Linda about some of the concepts, and I'm not really going to repeat any of this in great detail. When I, as a doctor, because the colleagues, as Annemieke said, were either mouse doctors or basic scientists, but when I, as a clinician, talk to patients that have a genetic muscle disease, it's often, and even 20 years ago, they would come up with the question, so can't you replace the faulty gene with the healthy gene? The concept of gene therapy is very simple. It's a lot simpler than, for example, exon skipping or explaining antisense oligonucleotides. So you replace a faulty gene with a healthy gene, and the challenge is how do you get your healthy gene there? And so you have heard from Linda and Annemieke about AAV. Now, the problem with AAV is that it's small. But what is important as well in this very easy, simplified schematic of the promoter and the functional gene, these are the components that companies change to make their products different. On the one hand, it's the viral vector itself, but then it's the promoter and the functional gene. And so, as I said, I don't need to really repeat any of this. What is important is that we have to talk about spinal muscular atrophy. Even though this is a conference about Duchenne muscular dystrophy, the headlines when it comes to gene therapy in neuromuscular diseases are actually with spinal muscular atrophy. And I think for parents in particular, and even though there are many colleagues here from industry, but nevertheless I want to spend a bit of time on this. Why has there been such amazing success in gene therapy in spinal muscular atrophy, and why is it so difficult in Duchenne muscular dystrophy? Now, funnily enough, if you would have asked Francesco Montoni or myself 15 years ago, we would have probably both put our money for gene therapy in Duchenne and would have said, spinal muscular atrophy type 1, you can't really treat that. It's too late. I mean, these kids are far too severe. There's fairly little that any therapy could really change, because these kids were going to die within 1 year, 18 months, all of them basically by 2 years of age. They are floppy babies, already showing symptoms in many cases at birth. So this is a baby with SMA type 1. And now you hear these news that you really almost have a cure. Now Annemieke did mention Jesus. I'm not quite certain. I mean, you could argue about him having cured people, but I mean that's for a different discussion probably. But even with SMA type 1, cure is indeed something where we need to be careful. What does cure really mean? But it clearly is disease-changing therapy for spinal muscular atrophy. So why does it work? Now, first of all, for those who don't know, but in Duchenne muscular dystrophy, our target tissue, where we want to replace the faulty gene with a healthy gene, is the skeletal muscle. And in spinal muscular atrophy, it is the motor neuron or the anterior horn cell here in the cross-section of the spinal cord. And there are a few really important differences between the motor neuron and the skeletal muscle. Neurons, as you know, will not replicate. So if you have a headache or a hangover and you think, oh, those neurons, they will not come back. And actually, you're born with a definite, defined number of neurons. Now, the good thing is they don't replicate, they don't reproduce. So once you have your gene of interest in that, what we call a post-mitotic non-dividing cell anymore, once that's in there, it's in there. Now, the problem with muscle is that muscle cells, on the one hand, replicate when you're young. For most of the people here in the audience, your own muscle cells replicate anymore, sorry. But in Duchenne boys also, so up probably until the age of about 20 or so, this is when replication and regeneration of muscle really stops, and it's more replaced by scar tissue. But in young kids, and in young mice as well, if you injure your muscle, they will generate new muscle. Now, this muscle still needs to grow, and muscle is also, in contrast to the motor neuron muscle cell, with many, many so-called nuclei. So this is where your DNA sits. So you don't have to just target one nucleus in a muscle cell, but many, actually a few thousand probably, in a mature long muscle cell of your femoral quadriceps muscle, for example. So there's a huge difference with spinal muscular atrophy and Duchenne muscular dystrophy. And I think what we currently don't know is, in boys or patients generally that grow, how do we dilute the effect of replacing the faulty gene with the healthy gene? But it's also looking at the disease onset and course. So in spinal muscular atrophy, we make the diagnosis often in infancy, or always in infancy in SMA type 1, even often in the neonatal period. So at that time, the immune response is fairly primitive. Our immune system gets cleverer the older we get. And again, once you have reached a certain peak, the immune response also declines again. But clearly when you are a neonate, your immune system is fairly naive, which makes it easier to expose the immune system of a neonate or infant with particles like viruses or proteins that they haven't seen. And of course, for a company, it also makes a huge difference in terms of viral load per kilogram body weight if you inject an infant or someone who's 50 kilos or 20 kilos. So there is also an economic aspect here in terms of production. I've listed outcome measure. That's a huge difference between Duchenne and SMA type 1. Now, those who know the field, in Duchenne muscular dystrophies, we have discussed outcome measures for probably the past 40 years, and we still continue to discuss outcome measures. In SMA type 1, there is a very effective outcome measure, and that's death. It's almost the best outcome measure that you can have. It sounds, of course, tragic, but if you can extend life, that convinces almost everyone to say, well, that's an effective therapy. So there was also an advantage, but even the severity of the disease, so if you can show that a baby that would normally never be able to sit can all of a sudden sit or can stand or walk, the effect size of your therapy is enormous. That's one of the reasons why, for example, in SMA, no placebos were necessary to be trialed. So none of the SMA gene therapy trials ever had a placebo arm. Would have been unethical. So the placebo or the phase 3 study was really only done for safety and making sure that what has been shown in one center can be reproduced. I already mentioned the target tissue. Now another problem is the gene size, and we heard from Annemieke and from Linda that the dystrophin gene is actually the biggest gene that we know in nature. And then protein expression, and again from the previous talks, you've heard that in Duchenne muscular dystrophy, many patients have never seen certain parts of dystrophin. In SMA, every baby that lives with SMA type 1 knows the full-length protein, otherwise they would not have been born. So in hindsight now, Francesco and I would have lost all our money, and SMA seems to be SMA type 1, the perfect condition for gene therapy. It's really something that we have learned. And Duchenne muscular dystrophy is very difficult. And the biggest challenge is probably that it's really, really big. When I talk to patients and patient groups and parents, I often do compare the genome with a library and the genetic code with letters. And actually, to understand how big the Duchenne muscular dystrophy gene, the DMD gene is, it is about 10 books in terms of the number of letters. So it's massive. And so we have already heard, this is why in terms of AAV, you can't put it into your normal big removal van or transporter. You need to have a different delivery, the mini dystrophin, and at least we know from nature that patients with Becker muscular dystrophy can have a fairly good function with a smaller dystrophin. And Annemieke used the analogy of climbing in the carabiner. We can also use this shock absorber here where you say, normal Duchenne and Becker. This chair clearly would collapse, whereas this would be less comfortable but would still work. And so this is a bit about the function of Duchenne and the dystrophin gene. Now, in this specific session, the focus was on what's in the pipeline for gene therapy, and then we'll have a later session about the latest in the clinical trials. So in the next session, we will focus on what are the trials that are currently ongoing, and what are we learning? Whereas in this session, we were going to focus a bit on trials that are upcoming, but are currently not recruiting or open in the UK. And so, for the probably next 10 minutes, 15 minutes, I'm going to focus on some of this. And again, we have now seen schematics of these, what we would call the healthy gene, and because of Duchenne and the dystrophin gene, we need to get smaller healthy genes in there, so they're not quite as healthy as people would hope, and that different companies have used different constructs. And so there are two trials that have already started, but not in the UK yet. One of them is by Solid Bioscience and one is by Genethon. The Sarepta and the Pfizer trial are currently ongoing, and we'll talk about those in the next session. Now for Genethon, all of those who have been here yesterday and listened carefully, I mentioned that there is already a natural history study taking place in the UK. That's the one where it's a bit difficult to recruit because of the inclusion criteria of the body mass index, that one of the inclusion criteria is to have a body mass index below 75%. And so, which is difficult with steroid treatment in Duchenne muscular dystrophy boys. And we said that this natural history study serves to learn more about the trajectories of the patients that will then be included into a gene transfer or gene replacement trial. And so this is the GNT0004 study from Genethon, the microdystrophin study. It is using an AAV8 vector, was actually developed at the Royal Holloway with George Dixon and Linda Popwell as well. So this is a vector that has been trialed in mice and then in dogs and was really very promising, and is now at a stage where the first patient has been actually injected in France. So this was a trial initially with Paris, London, and Newcastle. In France, first patients have now been injected. The aim of the study is clear. Again, you want to increase dystrophin expression. The age of the natural history participants was from 5 to 9. And so Francesco is the chief investigator in the UK for this study. Newcastle is also a trial site, but currently no patient has been recruited. This is something that's coming up in the future, so this is why it's listed as part of the future gene therapy trials. Now, the other one is the IGNITE DMD study, and many of you will have heard about this. This is from Solid Biosciences, and it's a trial that we thought would also come to the UK. The inclusion criteria are 4 to 17, so it's a lot broader than some of the other gene therapy studies. In contrast to the Genethon study here, it's an AAV9 vector that is used to deliver the gene. This is for patients that are also non-ambulatory, so with a broad age range, and ambulatory, and the protocols are slightly different. Solid has now closed enrollment for this trial, and to date 9 participants have received the treatment. the trial was also halted for a while because of some side effects. Now Solid decided to look on how these patients that have been injected will actually develop. So far, from what has been publicly discussed, is that the patients are stable at least, so there's no deterioration in motor function. And we expect that the trial will go into a next phase probably by next year. So that's the situation here. The construct, as I said, is different just more for the insiders, but the Solid construct also maintains the nNOS binding site. I think we don't probably know enough yet about the long-term relevance of some of these differences. And I think really competition is great in this field. This is another gene therapy trial from a company that has specifically changed the promoter and feels that their unique selling point is really that they have a promoter that allows the gene to be expressed in muscle at a far higher level than the promoters in some of the other constructs. So in preclinical studies at least, there's an increased expression in skeletal and heart using this SPC 5-12 promoter. And the vector, again, is an AAV8. So just a bit more information about the Regenix Bio study. So it's a phase 1-2, so very early on. And so it's expected that first patients will be injected early next year. Currently, the data is just based on preclinical experiments, but it looks very interesting. But I think what we have all heard yesterday and also this morning is that we have to do the clinical trial to really see whether what has been seen in animal models can also be translated to humans. So these are the gene therapy trials that are coming up in the UK. So it's, again, good to see that there are 3 in addition to the 2 that we already currently enroll into. And then again, I'm not going to talk about the principles of genome editing, but again, it's interesting. I mean, I'm often going to patient conferences and talk about clinical trials, and I remember that it was probably 6 years ago when I was at a conference. This was before we started to do gene therapy, and a parent stood up and said, "What about CRISPR-Cas?" And you thought, ooh, I would have never even thought about CRISPR-Cas in Duchenne at that time. But it seems everyone has heard about CRISPR-Cas gene editing and that this is the future. And I mean, again, you have to be extremely careful because we have heard this for so many things, and managing expectations can be very difficult. To really explain how it works is, again, a lot more complicated than replacing the faulty gene with a healthy gene, because it's almost a mixture of gene therapy and exon skipping as well. And Annemieke and Linda explains this in a bit more detail. So it's a very useful tool, but you actually need so far a toolkit with 2 components, this so-called guide RNA, and then the Cas, whether it's Cas9 or another one, CRISPR-associated protein, that's the scissor. So you need to have the guide for the scissor, where the scissor is supposed to go, and then the scissor itself. And most likely you may either need to deliver them in 2 different viruses. So it's still then going back to the transport. The tools are small enough to put them in the viruses, so that's good. So therefore, people are now trying to see, can we maybe even put them in one toolbox together and combine them. CRISPR-Cas has revolutionized science, but more as a scientific tool rather than a therapy so far. But I'm mentioning it because there's a company, Vertex Pharmaceuticals, that is looking at CRISPR-Cas in Duchenne muscular dystrophy. And so Vertex is a company that's specifically interested in rare diseases as well. They have been very successful in cystic fibrosis, and say, okay, Duchenne muscular dystrophy is one of our next big disease targets. They have already done studies in mice and in dogs, and again, it looks very promising in terms of dystrophin expression, but we have seen this for many other products. Annemieke mentioned that almost any therapy in an MDX mouse has shown benefits, so let's see. They are using an AAV9, so something that we also use in our other gene therapy trials. And I would expect them to come into the clinic next year. They will most likely target patients that are amenable to exon 51 skipping. As we said, it's a combination of gene therapy, delivering the toolbox by a viral vector, targeting the gene, but then doing a bit like exon skipping. Now the advantage, you could say, well, why is this supposed to be maybe better than gene transfer therapy? The advantage would be that you have a larger functional protein compared to the gene replacement approaches that use the viral vector with the dystrophin transgenes. So it will be interesting, and I think the clinical trials need to show how effective this is. Then this is a trial from Sarepta and Vision, and it's a phase 3 trial. And again, I'm listing this here as a future trial because we are currently not yet enrolling patients in the UK. So this is a study that, again, the study duration would be more than 2 years. The target is 116 patients, so it's very large. This is for also non-ambulatory patients. I think that's what makes it really interesting. Often when you go to patient conferences, patients feel, well, what's happening with the patients that are already wheelchair bound or that can't walk? And partly because in the past we have often emphasized that an important outcome measure is a 6-minute walking distance, which means you can only enroll patients that can walk. So I think within the community there has been a lot of learning. We know that there are unmet needs for also the patients that are in a more advanced stage that can't walk. And in terms of the therapeutic concept, there's of course no difference whether you can walk or you can't. The challenge is that your amount of muscle when you can't walk might be less, so we're not entirely sure about efficacy. On the other hand, if you talk to older patients, you know that if you could maintain their upper body function and their upper limbs, they would already say that would be amazing, and I fully agree with this. So the inclusion criteria are based on the North Star assessment. You can see that the age range is up to 18 years, and particularly the performance of the upper limb will be looked at. And so this is hopefully a study that will also come to the UK. And then very similar, there will also be from Pfizer a phase 3 trial that looks at patients that are non-ambulatory, So also here the study duration will be 2 years with long-term follow-up. I think for all of these studies there will be long-term follow-up. Approximately 66 non-ambulatory male patients are supposed to be enrolled. And again, the assessment will also include particularly assessment of the upper limb. So I think this is fantastic to see that broad spectrum of disease severity is going to be addressed in these future trials, and future means when they come to the UK. And just some closing remarks about cell and gene therapy. We haven't really talked much about the cell therapy. You hear about this again and again because cell therapy is in principle also gene therapy. It's just that you use, instead of a viral vector, you use a cell that carries the gene. And so with muscle, we nevertheless face the same challenge that muscle is the biggest organ that we have. 40% of our body mass are made out of skeletal muscle. So to target skeletal muscle by cell therapy is definitely a big challenge. So the challenges are for both of these approaches really around immunity. We heard this, upscaling of production, delivering to the tissue of interest, skeletal muscle, and ideally the heart, the gene size. And I mentioned the niche, it's particularly for cell therapy. Once you take a cell out of the body and put it in, this cell has changed its characteristics. We sometimes don't talk enough about other aspects that are actually challenges. One is pricing. These therapies are expensive to develop, and understandably, pharmaceutical companies want to make a profit as well. You have heard that for the SMA gene therapy, this was at the time the most expensive drug in the world. Payers like the NHS England need to, of course, agree on how much they are willing to pay, and insurance companies. So that's a parallel but very important discussion. Then I think the ethics as well, there are things that we really don't understand well enough. When particularly we talk about CRISPR-Cas, for example, everyone knows the gene scissor, but then if you would ask someone, well, are you happy to have a gene scissor in your cells for the rest of your life? And what is this scissor going to do? Well, we don't really know. So it's really informing the community about what first in human trials might mean. It's difficult to say. I just want to mention here one clinical trial with gene therapy in a different neuromuscular disease for a disease that is a congenital myopathy, and it's called myotubular myopathy. The gene therapy really works, but out of 24 patients in the clinical trial, 4 died. And so it's only in the trial that we learned about some of the risk factors. And it's really difficult to understand fully what is the risk benefit of a treatment and what would parents or patients be willing to risk. And we had a workshop, I think it was last year, organized in London by Emma Heslop and the Duchenne Hub, where we discussed this, and you realize that parents might be willing to risk more than actually the patients. Now, who's going to decide this? Very difficult. So I think this is also something that needs to be discussed, and probably we need to still see how safe some of these approaches are. But I think what is very encouraging is that we now see gene therapy becoming a bit more mainstream in rare genetic diseases. So in the neuromuscular field, we have now in Newcastle injected adult patients with gene therapy in Pompe disease. I've just mentioned the central nuclear myopathy trial. We are also involved in a limb-girdle muscular dystrophy gene therapy trial. So there is a steep learning curve for all of us. That was basically all about the trials coming up in Duchenne muscular dystrophy. And so thank you very much for your attention. And if you have any questions.
Probably hear me. Does anyone have any questions? No, that's okay. So I, again, have a few questions that have been sent. I asked a similar question yesterday about exon skipping. This was about the timing of when we give gene therapy. We know muscle health is important. We look at the progression of the condition with age. So in your opinion, I know the jury's out on what's best. Could you comment on that in terms of giving gene therapy and when's the best kind of time?
So the best time point of doing gene therapy is one of these, how do you say multi-million, really, literally, dollar questions. So the earlier you inject, the better it is in terms of targeting muscle. I mean, your muscle will still be there. And when I say early, let's say, you go into 4 or earlier. We know that muscle will not have been replaced by fat tissue at all. So in terms of good muscle, even though it's still Duchenne and damaged, that's the best time point. Yeah. But, then that muscle is growing and this person is growing. And so this goes back to this dilution effect. This is something that we don't know, and there are different views, and you speak to different investigators, and they would promote different ages. I have to say I don't know. I think from my perspective, and as a clinician and talking to patients and parents, if I could provide a treatment that would allow you to maintain or even improve, but let's say maintain, your hand and arm function, I think that would be amazing. So the advantage would be if you go into an older patient, let's say 15 years, normally they won't grow much more, so the muscle will not, regenerate a lot more and proliferate a lot more. So maybe an effect there would be fairly stable. But there's nothing that you can do with that therapy to make that patient walk again because there are no muscles left to target with gene therapy, probably. I mean, so therefore, I think there's no right or wrong answer for, this is the best age. I think we need to do these long-term, observations of patients, and then we'll know.
Completely agreed. Yeah. Okay. Okay.
I have a question regarding the side effects. Is it mutation-specific, the side effects, or is it related on different things on the genome of the human being? And is there a special study to check what's the relations of the side effects or maybe a new model to check? Because we hear that, on the mice and the dogs, there's no it's safe, but then on the human body, it's not safe.
So we will, in our next session about current gene therapy trials, we'll talk about safety in more detail. So there are probably also, based on Annemiek's talk this morning, mutation-specific side effects, depending on where you have the mutation, because you might have immunity against the introduced dystrophin molecule. And then there are more generic side effects against the viral vectors that we see across diseases as well, so not just for Duchenne, but in spinal muscular atrophy and in some of the other diseases as well. I think one thing that we definitely learn as well And I think that's what I wanted to show with the comparison to SMA, that we need to look at gene therapy disease specifically. And there's not, one concept that fits all. But coming back to your specific question more, so yes, there are certain mutations that have a higher risk. Why some patients respond stronger to the virus than others is just probably something that is depending on your immune sensitivity. But I think we are starting to really know the side effects in terms of liver problems or problems with your coagulation or your platelets, and I think and your heart. And what we are seeing is that the protocols of the companies are constantly adjusting to what we are learning. So we are starting to be able to monitor side effects more effectively, and therefore to also treat them earlier. So, you never know what you don't know, but I think that we are making good progress there.
How long do the effects last for?
Yeah, so again, a really excellent question, and it comes a bit back to the question, what is the best time point to inject? Because it directly relates to, well, how long do the effects last for? So you could say, well, maybe the best time point is to inject at 3 years of age, but the effects will only last for 6 years. Or it's better to inject at 15 years of age, and the effects will last for 15 years. So it's something that we really currently can't answer. I can't.
Okay. Yeah. Any other questions from the audience? No? Okay. Just another kind of general question as well. Sorry, I'm quite far away. In your opinion, what is the biggest challenge we have with gene therapy? I know Linda covered a couple, but what is?
Yeah, I think, Annemieke, you may have been asked the same question, wasn't it? So I think that the size of the gene for dystrophin is, for me, the biggest challenge. The other challenge is, you could say, related to this, we don't have a better transporter, the big removal van, where we can put the full-length gene into it. Maybe we need to work on this more, and of course people do. So I think this is, for Duchenne muscular dystrophy, for me, the biggest challenge right now. I think, you could treat Duchenne a lot more effectively if you could restore and re-express at a high level full-length dystrophin.
Sorry, follow-up question, but aren't you afraid if you express the full-length dystrophin that will trigger an immune response? Because
okay, that's the side problem then, okay? I mean, once we would be able to do that, I think, we would be in a lot better position. Definitely. And then we have to think about the immune response. So, considering that we do bone marrow transplantation and cardiac transplantation, and we can deal with an immune response fairly efficiently. We currently don't want to do this in Duchenne muscular dystrophy in the same way that we do this to, transplanted patients, because it might be overkill. Actually, it's also very expensive, so it adds to the cost, so on. But that's, for me, almost, a second problem that needs to be addressed, of course. But the challenge for me is currently delivering the full-length protein.
Are we sure we are targeting the right type of muscle cells? We are talking here, all the talk about myofibers, terminally differentiated functional muscle. Is it the right approach, given that dystrophin has been found in muscle stem cells, in myoblasts, having a role there? And these cells might be better, potentially. What's your view on that?
Yeah, and it's a very good point. So I think the question is really, is it sufficient to just target the skeletal muscle cell? And I mentioned this dilution effect, which would not be an issue if we also target the stem cell within the muscle, the satellite cell. And yeah, if you would, for example, ask Michael Rudnicki, he would say, this is exactly, I mean, Duchenne muscular dystrophy is a stem cell problem and not a, myofiber problem. So ideally you want to target both, I think, including skeletal muscle and ideally the brain as well. And, so yes, I think, in terms of targeting tissue in specific subpopulation of cells, everywhere where dystrophin is expressed.
I just have another question that I've met 3 different families. It's a bit more case specific, more about viral shedding after gene therapy. So there are certain families with 2 boys who have Duchenne muscular dystrophy, and only one of them is eligible for a gene therapy trial. I've raised this question with some of the farmer representatives as well. So, and one is eligible, one isn't. So do they have to be separated? Can you comment on the viral shedding?
So the viral shedding is something that the companies need to also provide information on before a trial will be approved in the hospitals. The hospitals need to know about this. And so we know that the viruses can be within saliva and body fluids, urine at the beginning, and also in stool for actually quite a long time, up to 4 weeks or even longer. Now, of course, these are sources for contamination, and therefore, you would indeed recommend that if you have a sibling that could be a future candidate for gene therapy with the same viral vector, that while there is viral shedding, avoid close contact. And I mean, families do this to different degrees, where you say, well, you'll live with the grandparents during this time, or you just have separated rooms, so everyone will actually get clear information about this with the clinical trials. If you are involved, there are instructions about how to deal with hygiene, how to handle, body fluids. So this is all part of the education that we do in clinical trials.
Is there anyone got a question? No. I've got one last question I asked the speakers earlier as well. As a clinical researcher for DMD, if you could have one wish that would make your job easier, what would that be?
Sorry, once again?
As a clinical research for DMD, if you could have one wish that would make your job easier, what would it be?
Okay. Clone Michaela and Emma, or no? God. Yeah. I mean, it would make my job a lot easier, and I think Linda mentioned this, if there would be more funding to, have sufficient staff as well. All of this is enormous, time-consuming as well, and we are all working really at capacity. We are very committed and, passionate about the clinical trials and delivering this, but whether it's, our research nurses, whether it's our clinical trial coordinators, physiotherapists, doctors, having more workforce to deliver what we want to deliver would be great.
I don't think we have any other questions. Thank you very much,
Thank
Volker.
you.
Thank you very much. Thank you.